Hello, I am presenting project number J-1031. For this experiment, I looked into decellularization and how the amount of SDS in the solution affected the stability of the scaffold. So, decellularization is the process in which the material from inside the cell and the DNA of the cell is taken basically from the cell, leaving behind a blank scaffold which can then be reseeded with new cells. So I got the idea for this experiment from a Pinterest post about a Tumblr post about a YouTube video where a guy made a meat berry. Of course, I had to go check that out. So I went to the link and I watched the video, and it turns out that the guy was decellularizing a grape and reseeding the scaffold, which was what was left behind, with fetal bovine serum, which is essentially cow cells. So I was really interested in this topic because what that meant was that it was a grape, but it was actually biologically meat. So it looked like a grape, but it was a cow. That was just astonishing to me. So I decided to make decellularization my topic for the 2021 science fair. Now, for this experiment, I had to look into practical uses for decellularization. So I saw in the medical field for organ transplantation. So the donor organ can be decellularized and then recellularized with the recipient's own cells, making the chance of rejection significantly less. Also, Dr. Andrew Pelling and his students have been working on creating spinal cords made of asparagus, and they, in fact, innovated decellularization as a medium for facial reconstruction by carving an apple in the shape of an ear and then decellularizing it, and then recellularizing it with the recipient cells and reattaching it, so as a base for skin to grow on. So for this experiment, I used immersion decellularization, which is the process in which the thing that you are decellularizing is immersed in a solution of a detergent and water. So I used sodium dodecyl sulfate and sodium lauryl sulfate. They're the same thing, just different names. So what that does is it pops open the cells. So that's very essential to decellularization. The thing about this method is, though, it takes about a week for the object to be fully decellularized, where there's another method called perfusion decellularization, which is where you pump the solution through the thing that you are decellularizing. And that means that decellularization takes a shorter amount of time, about a day actually, and it can be used for more complex structures. Immersion decellularization isn't really used that much, but for perfusion decellularization is used a lot for decellularizing organs, and because of the short amount of time that it takes. Anyway, for this experiment, I predicted, my hypothesis was that the more SLS in a solution, the more flimsy the scaffolds would be, but the less time it would actually take to decellularize. So that was proved, part of that was proved correct, because the more SLS in the solution, the less time it took overall. But the scaffolds didn't seem to have any effect from the more or less SDS in the solution. So in conclusion, my hypothesis was half right, but this experiment was very fun to conduct. Thank you for your time, and thank you for judging my video and taking time out of your busy schedules to look at my project.